In this video, we're going to look at the role that pH plays in the digestive system and how it interacts with enzymes in the digestive system. So in this part, you were asked to color the digestive tract according to pH, coloring in purple the areas that have a neutral pH and red that has an acidic pH. Well, it turns out that most of the digestive tract is operating at a near neutral pH. This is the conditions under which most enzymes like to operate. But as you can see in this drawing here, is that the stomach operates at a very acidic pH because of one particular enzyme that works best at a pH of around 2. Now, many foods are fairly neutral when you eat them, but not all. There are some that are acidic, like orange juice. Some that that can make your saliva rather basic, like some of the green leafy vegetables. But saliva contains buffers that can neutralize acids and bases. And that is because the salivary amylase functions best at a neutral pH. So the saliva is going to neutralize, whether it's acidic or basic, and neutralize the pH and allow that salivary amylase to work well as it travels down the esophagus towards the stomach. Once food reaches the stomach, our stomach is producing hydrochloric acid, and this is the ideal environment for pepsin to digest proteins within the stomach. If you recall, the enzyme is produced in an inactive form called pepsinogen, and it's only when it interacts with hydrochloric acid does it become the active pepsin enzyme that can begin the digestion of proteins. But the pancreatic enzymes work best at a neutral pH. So as that acid chyme leaves the stomach and goes to the small intestine, it is neutralized by the bicarbonate in the pancreatic juice. So it goes from an acidic pH to a more neutral or almost basic pH as it enters into the small intestine, and will stay that way throughout the rest of its trip through the alimentary canal. So this chart here is relative to part 7 in your packet, and it summarizes all of the enzymes that we've talked about here. We've got salivary amylase that's produced by saliva glands, and it's used in the oral cavity and continues to function as food travels down the esophagus towards the stomach, and it primarily digests carbohydrates, and it works at a neutral pH. Because pepsin works at an acidic pH, our gastric juice also has hydrochloric acid. This pepsin is produced in the gastric pits in the lining of the stomach, and then is secreted into the stomach lumen, or the inside of the stomach, and can begin the digestion of proteins into smaller polypeptides. After it leaves the stomach, then all of these enzymes here are enzymes that are produced by the pancreas and released into the duodenum of the small intestine. And they all work at a nearly neutral to slightly basic pH. And they include pancreatic amylase that digests carbohydrates. Peptidase, trypsin, and chymotrypsin all play a role in digesting small polypeptides that were created over here in the stomach, digesting them down into smaller and smaller pieces, even down into amino acids. Lipase digests lipids, and nucleases digest nucleic acids. So now it's your turn. Take a look at this graph. Try to analyze this data here. It's showing you the ideal pH curves for two enzymes that both digest proteins. One is pepsin, and this is the data that represents the ideal pHs for pepsin, and this represents a different enzyme, trypsin. So take a moment to think about what are the function of pepsin and trypsin? Where do they function in the alimentary canal, and what could explain the differences in function at different pHs?